And that goes for genomes as well. And there's some information in here on how to obtain the sequence experimentally, even from genomes. Genome sequencing is a really cool process. It's probably a lot more interesting than you think it is. But instead, let's jump into the sequence alignment. So this is a sequence alignment. Actually, this is a tiny fragment of a sequence alignment. This is the ribosomal 16S ribosomal RNA from a bunch of bacteria. In the ribosomal database, their alignment contains, the last time I checked, 1.6 million RNAs. And each RNA is about 1.5 kb long, but because of these gaps and things, the total alignment is about 5 times that long. So this is a huge database of stuff. But what is it? What does this look like to you? Does it look a little bit like a spreadsheet? It's a two-dimensional array, right? You have rows, you have columns. So what's in those rows and columns? Every row is a sequence from one organism, right? Or a single RNA sequence. And so I can't read it. I can read it on the screen. No. But each one of these is a 16S subunit RNA sequence from one organism. And there's a bunch of them. Each column is a homologous residue in that RNA. And the key word there is homologous. When you put a sequence in an alignment, and when you put a base in that position, like this A in that column, that's a statement. That statement is, you believe that that A is homologous to all the other A's in that column. What does that mean? Homology has to do with common ancestry, right? You know, your little finger here is homologous to the little finger of your parents and your grandparents, etc. Homology is actually a really complicated concept. Those of you who get into evolutionary biology, this finger is also homologous to this one, right? That's a different kind of homology. And it's homologous to your little toe. And your fingers are homologous to each other. All of these are different kinds of homology that are really interesting. They don't actually have a common ancestor, right? Because the common ancestor of my little finger and my father's little finger is a single cell from his developmental past. My finger didn't come from his finger, but they come from the same blueprint. Same is true for RNAs. Anyway, these columns are hypotheses that those residues are homologous to each other. And this is one of the advantages of RNAs over proteins in a sequence alignment. Because that homology, or at least structural correspondence, is a lot easier to sort out. We're going to talk a little bit about the relative advantages of protein versus RNA sequences for this. Proteins have one huge advantage over RNAs. Because RNA, what are the possible things in an alignment for an RNA? G, A, C, U, and gap. That's it. If you've got a residue there, you have C, A, G, and U, that's 2 degrees of freedom, not very much. Protein, you have 20 amino acids. A lot more information in each position in a protein alignment. But that's overcompensated by the fact that the assignment of homology is a lot better for RNAs. In the case of protein sequences, you use stuff like Clustal. How many of you have used Clustal or that? Yeah, okay. So what Clustal and related things do is they take a pair of sequences and they align them up in pieces to maximize the similarity between them. You're trying to make the sequences as similar to each other as you can. And then you build up on those pairwise similarities until you have a global alignment, in which the sequences in that alignment are as similar to each other as they can be in each position. Notice there's no mention of shared ancestry, structural details, or homology in that. You're just maximizing similarity. I'm not going to go through this. There's some tricks to aligning sequences by hand in the case of RNAs. you'll see on old exams that we spend a fair bit of time on this kind of stuff, and I'm going to de-emphasize that because you get a lot of it in genetics, right? And instead, we're going to talk a little bit more about trees. So you can look through this. There are lots of ways in which sequences are can be aligned or evaluated. So, for example, if you see, here you have a C and an A. That's a pretty hard clash because one of them is a purine and one of them is a pyrimidine. A purine to pyrimidine difference is a lot more important, a lot less likely than a purine to purine difference or a pyrimidine to pyrimidine difference. You'd like to see, so these A's here, at least the first three, are very highly conserved. And so this single difference here looks like a G is a big deal. As compared to other parts of the sequence that are highly variable, that's where differences have less meaning. You can actually score similarities based on these properties. But there's a better way to align RNA. So here's an RNA sequence structure. This is the RNA tRNA from Ralstonia eutropus. It's about 350 nucleotides long, and it's folded up into this nice compact secondary structure. This secondary structure can be used to drive your alignment. Because if you have a homologous RNA, here it is from the Thermobacter thermologicum. It doesn't take a brain surgeon to figure out that this A right here is homologous to that U right here. They're in the same place in the structure. Trying to align these two sequences by maximizing similarity will fail because these two sequences are only 24% similar. Now, you cut only four bases, that means two random sequences ought to be 25% similar, right? These are 24% similar. You cannot align them based on sequence similarity. But the structures are remarkably similar, and you can align readily based on these structures. Now, before someone argues that you can do the same thing with protein, the answer is you can. But it doesn't happen very often because proteins don't have highly defined secondary structures like this. Yes, they have secondary structure, but the positions aren't directly homologous to each other. They don't. If you have an alpha helix, they're not. The positions of the amino acids aren't the same within them. Whereas in RNA, you have direct base-base interactions throughout to help you register where everything is. Think about how RNAs and proteins fold. Proteins fold by collapsing into the so-called, molten ball sort of thing, right? And then the three-dimensional structure comes out, and the secondary structure is a direct result of the three-dimensional structure. And so the three-dimensional structure drives the secondary structure of proteins. The opposite happens for RNAs. In RNA, when it folds, the first thing that happens is it collapses into the secondary structure. All these helices form, they zip up in base pair. And then the secondary structure folds around until it locks into the correct three-dimensional structure. So the secondary structure drives three-dimensional structure in RNA. So these are very highly defined. You don't have to do any biophysics to solve the secondary structure of RNA. That's a bioinformatics problem. And I'm not going to get into how that happens. If you look in my lab, we're going to learn how to. So what does this actually look like? So this is what what an alignment of an RNA looks like. This is just a stem loop from a bunch of archaea. This happens to be P3 from RNA3. And you've got a bunch of stuff here that are annotations. The key is that if you've got, this is what they look like in secondary structure. And so this C and this G are homologous to that C and that G because they're the same number of base pairs out of the helix. The helix varies in length, but I can identify the homologous position. These ones don't have it, based on the location in the structure. And so when I align them, I can use these parentheses to mark base pairs. And so this parenthesis, whatever is in that column, base pairs to whatever is in this column. That's what you expect from homology, right? If this is homologous to this, and this base pairs to that, then this ought to base pair with that. Now that's a pretty high level of detail in a sequence alignment, far more than you can do with cluster or any of that kind of stuff. And the ability to create really, really high resolution alignments is the key to making phylogenetic trees with RNAs. Much of this is manual until you've got a great beginning alignment, and then their algorithms can use these seed alignments to fish out and include sequences using the same approach. And there's a program called Infer RNA nucleotide alignment that is used by the ribosomal database that you guys will use later on. And this can be done entirely computationally. With these parentheses notations, notice that the secondary structure of the RNA and the alignment are two different faces of the same coin. If you have an alignment and you know the structure of one sequence, you know the structure of all the sequences. And if you know the structure, then you can create the alignment. The two are really reflections of each other. You can't do that with protein very often. There is one case where you can do it with protein. If you've got three-dimensional structures of all the proteins you want to align, then you can do the homology model. You can superimpose two structures and line up in alignment the residues that are on top of each other. You can do this for a few proteins. Ferridoxin is a great example. If you don't really know the three-dimensional structure, you might be able to do a homology model from scratch. Take that sequence, fold it up into the same structure, do some energy minimizations, and hope for the best. But it's a lot of work, and it's almost never done. And so the trick is to solve the structure of the RNA and then use that structure or those structures to align the sequences. Sounds easy, and it certainly is maybe when you're working with stuff like this. Not so much when you've got that 3,000 nucleotide 23S ribosomal RNA, right? Actually, it turns out to be the opposite. These ones are harder. And the reason is the structures vary more than the ribosomal RNAs. Nevertheless, you can make great alignments. Phylogenetic trees are no different than any other computational process, right? Garbage in, garbage out. And so it's absolutely critical that your alignment be meaningful. I'm going to leave it at that. I've got some problem sets on these that you can work on. Go through the notes, go through the problem sets. We're going to do not this next discussion session. Maybe we'll do Devil's disease this time. Maybe something else depending on what the online students say. But the problem sets are tough. We'll do them not this Friday, but next Friday. All right? Those problem sets have to do with alignments. They have to do with secondary structure and going back and forth between structures and alignments, and then also generating trees. And again, I don't want to scare you with these trees. Some of these trees are pretty hairy. I know that you've already done that a bit in genetics. Certainly, it suffices for our purposes in this class. Instead, we're going to focus more on taking a look at a tree and understanding what information you can extract from it. Okay.
Mr. Brown, can you send out a PDF of all the labs?
Yes, and I have not done that yet.
Just something.
Yeah.